Okay, so here is a walkthrough of how to make a species tree. It's a little bit more complicated than I thought. It requires 2 additional pieces of software. So if you're having trouble and you can't do this, or get stuck somewhere along the way, I've got the software on my computer. You're welcome to come in and I can help you with it and do it for you. I'll provide links to both of the pieces of software that you're going to need also. You can also download them or you search for them in Google and find them that way. Okay. So the first one you're going to need is a piece of software called Mesquite. So I've downloaded and installed on my computer. You may need to update your Java software. It does require an updated version of Java to run. So it'll just take a minute here for this to load. And then what we're going to do is we're just going to make a unlabeled phylogeny, and then it allows us to edit it and move it around however we want to make the type that we want. So for this analysis, I have 4 species that are going to be represented. So I'm going to do file, new. You can give it whatever name you want. We'll save it there. That's fine. I'm just going to ask it if I want to replace it. This is just a temporary file, so I don't really care what it's named. So I'm going to do 4 different species in my analysis, and then I'm going to do it to make a new tree and just say make a tree using this source that I said was a default tree. So I don't really care about the relationships. I'm going to move them around anyway. So notice right now they just have a generic name on them. So we need to give them the specific names. So in this analysis, I'm doing a human. And you can use whatever names you want. Just make sure they match. You're going to have to put the same names in when you get to the mega portion, which I'll demonstrate here later. And was also in that worksheet that we did. So I've got a pigeon. I have a mouse. And then I have a fish. So again, generic. If I wanted more specific, that would be fine too. They just need to match later on. So for me, keeping the name simple is easier. Okay. So I come back to my tree view, and now one of the nice things about this program is you can manually edit the branches. So I can say, oh, I want my mouse more closely related to my human. So drag it over there, and it will connect them together. The pigeon is the next one, the fish is the next one. So that's the correct tree, but if it wasn't, I can make any sets of arrangements I want just by dragging these different branches to connect in at the different places. Okay. So once you have your species tree exactly the way you want it, we're just going to save it. So here we're going to go taxa and trees, save trees to file from the current tree. And one of the problems with this software is it only saves in one format. So that's why we need the second piece of software to switch it to the other format. So in this case, I'm just going to save it as my ops and species tree right here, .nex. So you want to put that .nex extension on it. It doesn't matter what the other things are for now, just as long as you know where it is. And I'm going to save it. Okay, hit OK. Saving is complete. Great. Now we're actually done with this software. So we've made a phylogeny the way that we want it, and then saved it into a file format that then can be used by this other program. So I'm going to shut down Mesquite. We no longer need it. The other program we're going to use is called Fig Tree. So we're going to open up this program. And again, I'll give you the links to these so you can download them. And then we're going to open the tree that we just saved. So we're going to go back here. It was Opson species tree. Here it is. Exactly the way you want it. This program doesn't allow you to manipulate and move things around the same way the other one does. That's why we need 2 programs. But this tree does allow you to export into a different format. So we're going to export it as a NEWIT format. That's the one that Mega needs. So we say OK. I've already done this as a practice, so there it is. We'll just replace it. So now we've got our species tree exactly the way that we want it. So the next step now is to get your gene tree. And this is using the Mega program and the stuff that we've already learned and gone over this semester, but I'll review it here for you if you want. So I've got a data set that I've built here. So I'm just going to open it up. And I just called it opsin.mass. So that's my data set. I'm going to align it. Everything's already been aligned here, so if I look at this, you'll see that I've got my protein sequences all lined up. And in this one, I've got 8 genes, 2 from each of the species. And looking at it, it's pretty obvious that I've got 2 different groups of orthologs, one up here and one here. You can see those quite distinctly. So this is a very simple, easy analysis. Yours may very well be more complex. Okay. So I'm going to make a gene tree. So all I need to do is data, phylogenetic analysis, and say OK. And then come back here to my main window and say, let's make a phylogeny. In this case, I'll just do a maximum likelihood phylogeny. Oh, sorry, I guess I did not do data, phylogenetic analysis, align sequence. Oh, I guess my alignment, something's up here. So let's just, it doesn't like my alignment. Oh, this is the version I saved before I did the alignment. That's why they look so different. So let me do my alignment real quick. So we'll do alignment. We'll just do the cluster version. This is a small data set, so it'll be very quick. So now we have a line version. It's a little bit better, and you can see more of the similarities, although there's still distinct differences between the two groups. Okay. So now we do data, phylogenetic analysis, and now that it's set, we can construct a phylogeny. And we've done this all before in previous exercises. So here we have our gene phylogeny now. Again, that's just a very quick version. You may want to do more analyses and double check them and see if they are congruent. But here we've got this. All I want to do is save this. Oh, sorry, it's actually not save, it's export. There we go, export current tree. Okay, so we're going to do this. And there it is. I'm going to save it now in a particular place. We'll just go to the same place we've been saving these. And it's in data sets. There we go. And this is my gene tree. So I'm going to call this opsin gene tree.nwk. That's the network format. So there we have now. We have our two complete trees, the gene tree that we made and the species tree. So now I can come back here to my main mega window. We'll shut down some of these others. And we can say, okay, I want to find our gene duplication events. So I'm going to click on this. So we now have all the files that we need. We have our gene tree, which is right here, opsin gene tree. Okay. Here we have the species name. Now this is where we need to make sure they're the same. So for every one of these genes, I need to give it the correct name for the species tree that I just made. So I'm going to do this, and I'm just going to manually enter them. This is probably the easiest way for you to do it. It might take a little bit if you have a lot of them. But so I just want to match each of these genes to the correct species. Mouse, human, pigeon, and fish. Okay. So I've got that done. Now I need to load my species tree. Same place, there it is, opsin species tree. Set the root for the gene tree, which is optional. You don't have to do. I think it was already rooted in the right place, but let's just go ahead and so you can see that. So here we've got opsin 1, 1, 1, 1. So if you don't know, which you very well may not, is then you can yeah, so it's already rooted to the correct place. But if you don't know, then you can just, it's optional, so you can just say skip. Okay, so we don't need to root the gene tree. In fact, it's probably best if you don't know ahead of time, if you don't want to bias the analysis, just to skip that part. Species tree was already rooted because that's the type that we brought in. Okay, so we're going to do launch our analysis now, and now here we've got this diagram. Again, it's a gene tree, but it shows speciation events in red and duplication events in blue. So this is a very simple pattern, a single duplication event ancestrally, and then those have been passed down, and each of those species still has both of those paralogous copies. Okay, so that's it. I will provide links to both mesquite and fig tree if you want to give it a go on your own. If you're confused or get stuck at any point, you can bring your aligned data set in here, and as long as you know what the species are in your alignment, I can help you do this, and we can generate this rather quickly and easily.